Today we are presenting the efficacy and safety of the oral PCSK9 inhibitor MK0616, a macrocyclic peptide, in the treatment of hypercholesterolemia. This is a phase 2b randomized placebo-controlled clinical trial which was presented at the American College of Cardiology 2023. So we know that elevated LDL is a causative factor for ASCVD and that a large proportion of patients fail to achieve guideline recommended levels despite statin therapy. We already have injectable treatments targeting PCSK9, resulting in significant LDL reduction and decreased ASCVD events. But there are access barriers and need for repeat injections, which have led to less than stellar adoption. An oral PCSK9 inhibitor may widen access and improve attainment of guideline recommended treatment goals. In the past, it was felt that it might be impossible to develop a small enough molecule because of the nature of the interaction between LDL receptor and PCSK9. With advancement in biotechnology using a macrocycle library, they were able to screen massive libraries of peptides in the order of 10 to the power of 14 potential compounds to find a peptide which was a good inhibitor. Then with medicinal chemistry, they were able to improve the molecule in terms of structure-activity relationships, pharmacokinetics, and stability. Now this gave us a precursor that was further improved. So this phase 2 multi-center randomized controlled trial of this molecule, currently called MK0616, looked at the dose response of 6 milligrams, 12 milligrams, 18 milligrams, and 30 milligram doses compared to placebo. So there was a 3-week screening followed by 8-week treatment and 8-week follow-up. The objective was to evaluate the effectiveness versus placebo on percentage change from baseline LDL at week 8. The hypothesis being that at least one of the 4 doses is superior to placebo on percent LDL reduction from baseline at 8 weeks. So these were male and female participants ranging from 18 to 80 years of age with a range of background statin therapy and a range of ASCVD risk with corresponding LDL criteria. Approximately 49% were women, 55% had diabetes, and about 35% of patients were non-white. About 39% had clinical ASCVD and 56% had intermediate to high-risk ASCVD. The average LDL was 119.5 milligrams per deciliter, which is about 3.1 millimoles per liter. So as a reminder, the primary endpoint was percent reduction in LDL. Here we can see that all 4 doses had significant reductions in LDL, which range from 41.2% at 6 milligrams, 55.7% reduction at 12 milligrams, 59% at 18 milligrams, and 61% at 30 milligrams. Near complete efficacy was achieved by 2 weeks and persisted over the 8-week treatment. Secondary endpoints of ApoB and non-HDL were also consistently reduced. In terms of protocol-defined LDL goal achievement, it was achieved 80% of the time at the 6 milligram dose and 91% of the time with the 18 and 30 milligram doses. Regarding safety, there were no organ-specific issues. There was really no significant differences compared to placebo. There was 1 death related to motor vehicle accident. There was no dose-dependent discontinuation rate difference. Discontinuation rates across the board were quite low. So in conclusion, this phase 2b randomized controlled trial in a diverse population of hypercholesterolemia patients demonstrated that all doses of MK0616 had statistically superior reductions in LDL-C versus placebo, with up to 61% placebo-adjusted reduction from baseline. Results were consistent across all subgroups. Improvements in ApoB and non-HDL-C were also consistent. MK0616 was well tolerated with no overall trends in adverse events across treatment groups. The authors conclude that these data support further development of the MK0616, which is an oral PCSK9 inhibitor, may improve access to effective LDL-lowering therapies and improve attainment of guideline-recommended LDL goals aimed at reducing cardiovascular risk.